Hello, I am Beth Ringwelski and I'm a student at North Dakota State University in Fargo, North Dakota. I'm in the Biomedical Engineering Master's program, and here I will be talking about our work dealing with label-free cell purification following electroporation. This work was an extension of a previous study done in our lab which dealt with the purification of live cells from dead cells using a microfluidic device. In that study, live cells were able to be successfully purified from the dead cells using dielectrophoresis. The dead cells stuck to the electrodes while the live cells were able to flow through unharmed. The hopes for these experiments is to use the purified and electroporated cell samples for immunotherapies. Compensatory proliferation is a process in which dying cells are able to cause the proliferation of adjacent cells or existing cancer cells. This damage could negate the positive effects of immunotherapies, and this is what we are trying to prevent. Here we wanted to see if time was a factor in this purification process. We wanted to know how long a cell sample was viable following being electroporated and before running through the microfluidic device. We tested 8 different time points ranging from 0 to 210 minutes. Here we have a diagram of the microfluidic device setup that was used during the experiments. The cell samples that were used were pumped through the device starting at the inlet on the left side to the outlet on the right. We used a mixture of 50% live and 50% dead cells to test the accuracy of the device. Jerkit cells were utilized due to their likeness to T cells, which are used in immunotherapies. The settings that were utilized on the device were 5 MHz as the frequency, 3 volt peak-to-peak as the voltage, and 7.5 microliters per minute as the flow rate. These numbers were perfected during the previous study done by our lab. The dielectrophoresis, or DEP, forces affect the cells when they are in a non-uniform electric field, similar to what they were experiencing while mixed with the buffer on the electrode. This is what causes the dead cells to stick to the electrodes while flowing through the microfluidic device. The COMCELL simulation shown at the bottom of the figure shows the electric field gradients which are required for creating DEP forces on the cells. The results of the experiments can be explained by looking at these two graphs. The one on the right is to prove that our microfluidic device was successful in separating the live cells from the dead cells. It shows the percent of live cells at the inlet of the device compared to the outlet at different concentrations of live to dead cells. The graph on the left features the results that we were looking to obtain for this specific experiment. Again, we wanted to know if time was a factor when completing these purifications with the microfluidic device. The percent of live cells at the inlet compared to the outlet were above 90% for the first hour following electroporation. After that first hour, the percentage of live cells at the outlet started dropping as the duration of time increased. The microfluidic device was able to filter out the live cells from the dead cells with close to 100% purity. This is due to the combination of DEP forces and the morphological differences between the live and the dead cells. The dead cells have a sort of burst structure with more rigid edges. At the frequency of 5 MHz, the live cells could be experiencing a weaker DEP force, which allows them to flow through easily, or the dead cells could be experiencing a stronger DEP force, trapping them to the electrode. With this extended study, we have found that the cell samples should be purified within 1 hour of electroporation. After 1 hour, the cells in the electroporated samples start to die and experience morphological changes. These changes can hinder the effectiveness of the DEP forces while being flowed through the device. However, if the cell sample is used within 1 hour of electroporation, the live cells will be successfully purified from the dead cells using the microfluidic device.